Hello everyone, welcome to today's live broadcast Chemically Modified mRNA as a Biologic, developing an analytical framework for success. I'm Chris Spivey, Editorial Director of Pharmaceutical Technology, and I'll be the moderator for today's event, and we're extremely pleased to bring you this webcast presented by Pharmaceutical Technology and sponsored by TriLink Biotechnologies. At this point, I'd like to share a brief statement from our sponsor. TriLink Biotechnologies, part of the Maravai Life Sciences, is a CDMO helping life science leaders and innovators overcome challenges in the synthesis and scale-up of nucleic acids, NTPs, and mRNA capping analogues with scale-up expertise and unique mRNA production capabilities, including its proprietary mRNA capping technology. TriLink continues to expand its cGMP and general mRNA, oligonucleotide, and plasmid manufacturing capacity at its new global headquarters in San Diego to support therapeutic, vaccine, and diagnostic customers. We have a few important announcements before we begin. Our webcast is designed to be interactive, so we very much encourage you to ask questions during this event. You can submit questions by typing them in the Q&A box, which can be found at the bottom of the video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player. The slides will advance automatically during this event, but if you have any technical problems viewing or hearing this presentation, please click on the question mark help widget at the top right of your presentation window. And now I'd like to introduce today's speaker. We're delighted to be joined by Khalid Yarmout. Khalid is the Senior Director of Manufacturing and Quality Control at TriLink Biotechnologies, where he oversees all analytical aspects of method development and validation to product release and stability to support regulatory filings for both small and large molecules. Prior to TriLink, Khalid held various positions in quality control, research and development, and manufacturing, where he supported a number of drug substances and drug products, both small molecules and biologics, from clinical phase to commercial. These include diverse experience and expertise ranging from discovery to manufacturing with Fortune 500 firms, as well as small entrepreneurial businesses in the areas of synthetic, analytic, colloidal, surface modification, protein and antibody modification and purification, covering both manufacturing and analytical testing and characterization. So it's a great pleasure to hear your presentation today, Khalid. Could you please get us started?
Thank you very much for the introduction, Chris. Much appreciated. So today I'd like to focus on the talk of chemically modified mRNA as a biologic and how we went about developing some analytical framework for a successful characterization of mRNA. I'd like to start with a brief introduction to mRNA. Probably a lot of you already know what mRNA is. And now it's a therapy with a newer technology engineered to mimic processed mature nucleic acid transfer presented in eukaryotic cells. The drug substance is made by a traditional in vitro transcription process of a target gene from a linearized DNA template. The resulting drug product contains the mRNA of the gene of interest and the required regulatory elements for translating the gene of interest, which include the untranslated regions of the 5' cap and the poly A tail. As I go through the presentation, you'll understand a little bit more, or probably already know the importance of the capping and the poly A tail in the process of translating. Without those, there will be no translating, and the mRNA would not produce the protein of interest in the cell for any therapy. in terms of quality and regulatory challenges for mRNA, first of all, mRNA before COVID, it was mainly used as a diagnostic and IVT tool. There was some work in terms of mRNA therapies for therapeutic use, but it was not very well known, but it was in the background being done. So in general, based on the diagnostic and IVT, the mRNA analytical methods, they were qualitative or at best semi-quantitative. And with mRNA moving into therapeutics right now as a drug substance and a drug product, the requirement or the need for a true analytical method that are very quantitative with a lot of sensitivity and specificity is needed. Also, the number of regulatory precedents is limited, but the diversity of mRNA-based application has become very, very broad. And, from a regulatory standpoint, there's 2 COVID vaccines that have been approved by the FDA recently. And those are the main mRNA-based therapeutics that have been approved by the regulatory agencies. Also, the challenge that people have is the lack of harmonization across multiple global regions. And that showed during the launching of the COVID vaccine, what Japan, EU, US, Singapore, Africa, in terms of the regulation and the need. And that became a slight barrier for analytical development for characterization of mRNA, since each agency required certain things a little bit differently. Now, in addition to regulatory uncertainty, there are many manufacturing challenges that are, that need to be controlled. And one of the main ones that is really important is the double-stranded RNA, and the reason for that, it's proved to be immunogenic. So the regulators look very carefully at that the double-stranded RNA is cleared, and it's not in your final product. So a lot of people ask, how do we classify the mRNA? And there's no regulatory body, framework to follow or something like that. But if you look at it, mRNA, through its process, it could be considered as a biologic, and it's right now classified as a biologic. So if we say, if we go with this classification as a biologic, then from regulatory standpoint, we have a huge history that we can follow and piggyback on, or use to leverage. Because we do have over 50 years plus in antibodies that have been approved, and vast array of products have been filed and approved following these requirements. So there is something that we could follow. Also, there are multiple process and quality attribute similarity between the current biological molecules, such as antibodies, and the mRNA drug substance. As for drug product as a sterile product, we can follow some previous drugs that have been approved using lipid nanoparticles. One such product is sold, which bupivacaine as a painkiller is encapsulated with lipid nanoparticles. And that has been approved product for quite a few years, where they inject it at the site of the surgery to manage the pain, which is a sustained 72-hour sustained release particle. So we can follow these requirements for the lipid nanoparticle, what was needed to approve that product as a sterile injectable using lipid nanoparticles. So if we look at the process similarities between mRNA and biological molecule, this will paint the picture of how similar they are. So for both, the first step is plasmid preparation, transformation. You select the clone, you establish your master cell bank and your working cell bank, and that goes for both. Then it's followed by the following step. Well, for proteins or for antibodies, you do a cell amplification and cell expression. Now for mRNA, you do a pDNA cell amplification and plasmid expression. So it's kind of, again, follow the same processes, they're pretty similar. There's differences, but it follows the high level. It's very similar. So for antibody, the downstream, you do isolation, multi-purification depending on your antibody, and if it needs to be refolded, and you get your protein as the drug substance to be reformulated as a drug product and used for therapies. For mRNA, you do a plasmid purification, you linearize it, you do your IVT reaction, and then you do a purification and you get your mRNA. So overall, there's a lot of parallelism between the two processes. And that actually helped us quite a bit to see what was done for antibodies and how can we leverage or use the same logic to apply it for mRNA and have a successful filing. So in summary, the quality process controls requirements are expected to be similar for upstream and downstream for both processes. You do have to do cell bank characterization and stability for both master cell bank and W cell bank. If you did any biologic or antibodies in the past, that is a major requirement that the regulatory bodies ask for. Control of raw materials, that applies not only to mRNA and biologic, but also to small molecules or anything that you made to be a drug substance. Clearance of processing agents, that is also applied to biologic and small molecule, where you need to look at residual protein, residual DNA, in this case, any residual NTPs that you have left over, or any solvents if you used it in the process. This is going to be further formulated for an injectable, so clearance of endotoxin and microorganisms is important. And all of this needs to happen in a controlled rooms where you have environmental monitoring program of the manufacturing facility. So you can see all these requirements that apply for antibody. It's the same thing that you can follow to be successful on your mRNA program. So next, I really want to step back a little bit from the analytical and the process and focus a little bit on the control of raw materials. This is becoming an issue in mRNA that a lot of people trying to establish a GMP program for making mRNAs, there is some struggle with the raw material. How do we know the raw material is GMP? There is not that many USP grade material and so forth. So, and with the growth of the mRNA for use in therapeutics, there's so many programs, and the vendors have not caught up. Raw material vendors have not caught up with a lot of programs to provide GMP material or to be up to that standard. So in terms of the control of raw material, I'd like also to narrow the focus on the NTPs, modified NTPs, and capping agents. So as you can see from the cartoon on the right, for your coding region, you need the NTPs or modified NTPs such as pseudo-U, N-methyl pseudo-U, or 5-methoxy-C. And in our case, we use capping agent, and this is one of the representative of the 5' cap, a clean cap reagent. So, and TriLink actually make these in a GMP environment, and we provide those as a GMP material. But what are, in terms of attributes of the building block, what you should be looking for? Well, GMP grade. You really want it to be synthetic, so it's free from any residual biologic, and all of our NTPs, modified NTPs, and capping agents are all made synthetically. You want it to be scalable to support life cycle of the product, and you also want to make sure your specifications are representative of the synthetic process. Now, what I mean by that is, if your synthetic process, let's say, use ethanol, you really need to check your residual solvent for ethanol, and you have to have it as a specification. You cannot just say, I cleared it. I'm evaporating the solvent, and it all should gone. So that's what kind of make it a GMP grade, and specifically, to support filings and so forth. So some of the GMP capping agent are, for example, we have a clean cap. It's a triphosphate to the left. It's chemically synthesized, Currently, it's used in over 600 mRNA clinical programs. So there's because it actually have a high efficiency of capping and improve translation to make the protein. And also a clean cap is already in billions of mRNA COVID-19 vaccine doses worldwide in so many different arms around the world. Your other NTPs is the GAC and the uridine. And recently, TriLink launched a GMP N-methylpseudouridine, which I believe that was the first available of true GMP N-methylpseudouridine for the use in mRNA. And it's a large scale, it's synthetic, and it's and I'll show you later on what do we mean by GMP. So there are certain advantages for using clean cap. If you see the schematic on the right, it's a process step comparison of clean cap to enzymatic capping. So for clean cap, it's a one-pot transcription. You do a purification and your final processing, and you got your mRNA. In terms of enzymatic capping, you do the transcription, you diafiltrate or clean it up, and then you do the enzymatic capping. Then you do another purification to get your final processing. So it's a high yield, so it's 3 times the yield of capped material compared to traditional co-transcription capping methods. It's a single pot reaction, so you remove 10 enzymes from the process to shorten manufacturing time. And not only that, you don't have to worry about residual biologics or large residual proteins that you need to clear from your process. Clean cap is synthetic, so there's no residual biologic in it as compared to enzymes. Proper capping, the clean cap will produce a cap1 structure for the most biologically active and least immunogenic mRNA, and also savings, more robust and cost-effective than enzymatic capping. Now, I really want to pause a little bit on this one, because if you want to look at the price of enzyme and price of clean cap, some people can say, I can make my enzyme in-house, so it's cheap. Maybe, but you have to look at the whole process. So you're saving a lot of manufacturing time, a lot of processing reagents, and so forth. So if you want to compare costs, it should be compared, as the whole process, not just as the starting material. So an example of GMP characteristic of a modified NTP, as I mentioned earlier, TriLink just recently launched N-methylpseudouridine as a GMP grade. Again, that's synthetic. There's no residual biological in it. And also the synthetic process controlled for mono and diphosphates as byproducts. And as you can see, I've listed all of the example of test method to support the specification. And all of these are related and cover the process. So, your parameters, your usual thing, the visual, pH, concentration, we monitor purity by HPLC. And not only that, we also have an impurity profile that we can monitor throughout the life of the product through stability and additional manufactured lot. We look at purity for pyrophosphates by P31, identity by mass spec and proton NMR. So as I mentioned, this is a triphosphate. As a triphosphate, we do an ion chromatography to determine the content of sodium. So we ensure that you only have your counter ion of sodium and not other excess sodium that could interfere with your reaction. Residual solvents by GC, we'll make sure its nuclease assays are free of RNAs and DNAs, so it does not show up your mRNA when you make it. And again, we monitor for endotoxin and bioburden. So what does that mean, GMP? Well, we can support this in GMP by controlling the raw material that's coming in for the synthesis. We actually do qualification batches and the process qualification to make sure it's reproducible with controls, process controls. And also in terms of the release criteria, it's all done, to make sure we are accounting for all the processes, all the impurities from the processes. So with that, it's practically a whole package that you can see to say this is truly a GMP material made under control record and a controlled process. So now we'll shift gear to quality requirements, and from raw material. So the quality requirement similarities for biologic and mRNA are going to be the same. For drug substance, you're expected to do identity, content, characterization, integrity, purity, functionality, / potency, safety, and general methods that you need for downstream processing to make your drug product. So this is a quick representation of the mRNA structure. As you can see, you have your capping agent, then the 5' UTR, the initiation coding and the coding region in the open frame, and the coding region is the sequence that you want to do that include NTPs and modified NTPs, and you bracket it then with the 3' UTR and the poly A tail. Now, the poly A tail is very important for stability of mRNA. You don't have the poly A tail, it will impact the stability of the mRNA. The capping agent and the mRNA are very crucial to the binding to the ribosome in order to do the translation to get the protein of interest for specific therapies. So if you don't have your capping and the poly A tail, you will not get any translation. So, we've followed a similar approach to antibodies, or actually small molecules, to develop an analysis of mRNA. Personally, I'm an organic chemist by training in my early years, and I've been using that same concept for biologics, and now, using it also for mRNA, where when you synthesize a small molecule, you have to characterize it. And the characteristic is not only did you make the right molecule, do you have the right functional groups? So we look at the whole structure of a small molecule and start determining what method you use to determine certain aspect of that molecule that's present. So now if you apply that same methodology to mRNA and you look at the whole structure of mRNA, so what can we do with an intact mRNA? So integrity and purity, and that would be the last thing that I'll present regarding the methods. But also, what can we do as a full length? Well, as a full length, you have to do identity. So currently, identity is done by sequencing. So you can use a Sanger sequencing or NGS. You can do some RT-PCR, if you design the proper oligos for all of that. And the other thing could be fingerprinting, and companies starting to do some of that. And that follows the concept of peptide mapping in antibodies. That's been an acceptable ID test by the regulatories, where you have a certain specific digesting of the antibody, and you ran it by LC-MS or LC, and you determine the fragmentation, and you know what each fragment is in terms of the amino acids, and that becomes you're mapping the peptides that you're digesting, and you compare that to a reference standard, and you can say that that's good for identity. The second thing is functionality. So how do we know we're getting the right thing? So this could be done two ways. One is, and I'm calling it functionality for in vitro translation, and then with a Western blot, to show that you made the correct protein that bind to the specific molecule or antigen for the therapy. So this is really a functionality, not really potency. For a true potency, this, I would say, you save that for the drug product, which will be a cell-based assay. So those are very complicated. The difference here between mRNA and antibody is, with the antibodies, your DS and DP have the same molecule in a way. They bind to the cell the same way. So you can do a cell-based assay for functionality for both DS and DP. For mRNA, it really doesn't need to, as long as you know functionality that your mRNA is fine, then once you encapsulate it with the LNP, if you chose that route for delivery, then the cell-based assay would be a true measure of functionality because for potency. So it'll cover both functionality and potency because you're translating the right protein that binds to the cell and see how the cell behave. Next is like we said, the capping is important. So we do have a capping efficiency, and for TriLink, we developed an LC-MS method that's actually could be qualified and semi-quantitative to determine the capping efficiency. Same thing with poly A tail. We developed an LC-MS method for that, and we can characterize some of that by reverse phase HPLC. In terms of impurities, you really want to look at your residuals. And so for this process in particular would be residual DNA by qPCR, residual protein. We prefer nano-orange for sensitivity, but some other labs use BCA. For residual NTPs, so you put all these nucleosides to make your mRNA. Did you consume all of them? Were you able to get rid of all of them, or there's some remain? So we were able to develop an anion exchange HPLC method to determine low levels of NTPs in your mRNA. Solvent is GC, USP 467 is the standard. And double-stranded RNA, you can either do immunoblot or ELISA. And for safety, your endotoxin and bioburden per USP. For content, we use UV or IPRP HPLC, but some labs use RT-PCR or RT-qPCR. And for others, appearance pH, you can add conductivity, osmolality, depending what do you need to as a characteristic to go further downstream to make your DP. And for integrity and purity, your gel electrophoresis, CE, size exclusion, or IPRP HPLC for purity. So following that concept, as you can see, we constructed a host of methods for each section of the mRNA to be able to characterize it and determine the quality and analyze it properly. So I want to switch gear now to go over some of the analytical methods that we've developed and used across multiple. First, we'll start with overall, there's a lot of companies have developed various analytical methods to characterize mRNA. And recently, USP provided a draft set of analytical methods for mRNA. And currently we are in collaboration with the USP to contribute to that based on the work that we've done. So for today, I'm going to go over 4 different methods, which is the IPRP HPLC. We'll look at purity, stability, and if it's stability indicating, content, and a little work on poly A tail, capping efficiency, poly A tail, and residual NTPs. So for the IPRP HPLC, this method developed and can be utilized to determine purity and concentration. For the work that we've done, TriLink has been making mRNAs for a long time. So we do have a large catalog of mRNA available. And we took that to our advantage, and we selected for this study 5 different lengths of mRNA to see how robust the method and how it could be applied to different types of mRNA. We carried that across all of the method developments what we've done. And as we go forward, I would explain how important the constructs at the 5' UTR and the 3' UTR, that if you keep those the same, you can do quite a bit, but if you keep changing those, you have to keep developing certain aspects of the method. So this is an overlay chromatogram of the 5 different mRNA. And as you can see, we can see a separation between different sizes. Now, but also you can see that, for example, between 1929 NTs and 3420 NTs, you don't see that much separation. I just want to clarify here. This is the main difference between this and CE. CE will tell you your molecular weight. It's not going to tell you other impurities. But with IPRP HPLC, your sequence, all the way the sequences and all the molecules that's there will interact with the columns. So this is more based on sequence separation, not necessarily size separation. And you can see a little bit more of impurities. Those are not very clean, and that was done on purpose to show that we can see other impurities that, for example, frag analyzers using capillary electrophoresis, it would not see it. You'll just see one smear. So with that, we were able to differentiate in terms of the sequence type and the sequence length. And then we wanted to look if this method is stability indicating. That way we can use it to determine the stability of mRNA. We did some heat degradation, and if you look at this by 60 minutes at 90 degrees C for a wild type F-Luke mRNA, it completely degraded at 60 minutes. Same thing, we did a base degradation for the same mRNA at 0.025 molar sodium hydroxide around pH 12, and within 30 minutes you can see a complete degradation. Same thing with acid degradation at 0.025 molar HCl for the same mRNA at around pH 1. So I do understand, you might say, well, those are harsh conditions between the temperature, pH, and the pH. Well, that's done on purpose, so that way we can forcefully degrade it and see that if something happened over time in storage, we will be able to see it by this method and determine what the purity over time. Now, can this method separate if you have 2 different poly-A tails? So again, this is really interesting because if you take we took a Cas9 wild type mRNA and we made it with 2 different tails, one at 120 and one at 180. If you put those together in a capillary electrophoresis, you're not going to see the difference. The 40 A's are not large enough of a mass to separate them, but clearly you can see that by IPRP HPLC. And this is an overlay, but again, we did some co-injections and you still see 2 peaks separated, not necessarily baseline, but again, we can change the gradient and a little bit of the chromatography condition, and we can get a full separation. But for this purpose, we're locking the method with a certain things that can we see all of that with this as is without changing parameters for everything. So with this method, you can, if you don't have the right tail, compared to a standard or what you're looking for, you'll be able to see it. Now, the other question that we asked is, could this method also differentiate between tailed and untailed mRNA? So again, if you have an 80 poly-A tail on your mRNA, that's, let's say, about 2,000 NTs, your untailed mRNA by capillary electrophoresis or frag analyzer in this case that we tested, you're not going to see the separation. So you really cannot determine if you have any untailed mRNA in it. But with the IPRP, what we've done is we took the same thing, wild type F-Luke. We made the untailed mRNA, and we did a spiking study here. We went from 0.5% to 50%. And you can see how, if you take a look at your 50% untailed, the area under the peak is exactly the same. So it is quantitative. And now if you take your untailed mRNA and do a standard curve, which we've done, and you can determine the LOQ and LLD for that specific sequence. And with that, you can have a method not only to measure the purity of your intact mRNA, but also how much there is of your untailed mRNA is present. So this is provided to be a very good tool, but I'd like to caution everyone here that this is really sequence specific. So for example, if you used uridine, you're gonna get a different retention time than if you used pseudouridine or N-methyl pseudouridine, or if you use C on 5-methoxy-C, you're gonna get different retention time. And this shows that, you know, your sequence, it's sequence specific because it's interacting with the column. Now, in terms of the method, you know, we use the same fluke, and we did a standard curve. As you can see here, we were able to get a good correlation. So this method, you know, we did specificity in terms of the performance tailed versus untailed. It's linear between 0.001 mg per ml to 0.3. We did accuracy by spike recovery at 80%, 100%, and 120 nominal, concentration of 0.2 mg. And for precision, the percent RSDs were below 2%. So the questions always come, how can we cut down test release? And that's a question I get a lot of time. And people say, can we not use this method? Can we not do that? I usually take a different approach, and my approach is how many things I can get out of one method. So in this case, if I run the IPRP method, I can get concentration, I can get purity, I can look at the impurity profile. It tells me a little bit about the poly A tail. So I get a lot of information from one method. And, you know, my philosophy is this is how you can speed things out of QC, by selecting methods that can tell you multiple things. So in summary here for the IPRP HPLC, it's a method that will distinguish between mRNA of different sizes. It's stability indicating. It distinguishes between the same sequence with or without poly A. It distinguishes between the same sequence with different poly A tails, and it could be a quantitative method for mRNA contact. And you know, you can determine your purity. Now, I'd like to move now to a quick overview on the capping efficiency. And again, as I mentioned before, the 5' UTR is very important for this method. So if you change your 5' UTR between sequences, then you're going to have to change something about the method. So the way the method is set up is we constructed a DNA enzyme that anneal to the 5' UTR, where the DNA enzyme will cleave the capping agent, and it will keep the rest of the mRNA intact. We'll do a phosphate treatment and then analysis by LC-MS. So from that, you can calculate your relative capping efficiency by taking the sum of mass intensity for identified 5' cap species and the total sum of mass intensities of identified 5' capped and uncapped, and multiplied by 100. So this is an example of our Fluke mRNA wild type. And as you can see in the chromatogram, the 13-mer capped one and the 13-mer uncapped. And in this case, we got 98% capping efficiency. Now, we took that method and applied it to all of the mRNA constructs that we tested in the IPRP for different lengths, to show that the lengths really does not impact quite a bit if you have the method, if you have the same UTR. And we had all the results are over 95% capping efficiency. And again, that's using CleanCap. Now for poly A tail characterization, one of the methods that we did is you take the enzymatic dissection of mRNA using RNase A and T1, and that practically chops down all your mRNA but keep the poly A intact. And you know, after sample treatment and isolation, what you see is the the chromatogram on top where you see your digested mRNA fragments and the poly A tail. And you take that peak and do a deconvolution on the poly A tail. And as we all know, this is going to be a distribution, not one number. So in this case, this is supposed to be 120. And we're getting a distribution between 122 to 126 with an average of 124. So again, the poly A tail going to give you a distribution. And from there, you can get an average of what your poly A tail in your mRNA. So in summary, the poly A tail method, it's a multi-part procedure. You have an enzymatic digestion of main mRNA using RNase, enzymatic treatment to move the 3' phosphate, work up the reaction, and LCMS analysis of the undigested poly A tail segment to estimate the average length and distribution of the length. So what we found is that method have a limitation to 148 poly A tail. You get a distribution curve / value of poly A. It is qualitative to semi-quantitative method, but it's good for characterization. And it's in terms of release, it provides the mean to characterize an mRNA material. And you can compare the quality of samples and determine batch-to-batch reproducibility based on that, which is very important. So the next step, the fourth method I was going to speak to today is residual NTPs. Now, you take the sample, your mRNA sample, you just do a spin column on that, you take the supernated, and you analyze it by AX HPLC, anion exchange. So here is a residual NTP and capping agent chromatogram representative. So you can see your C, your A, N-methoxy pseudouridine, U, 5-methoxyuridine, G, and the CleanCap AG agent. So we can get a total separation. And from there, we did a lot of spike recoveries. We did a lot of LOD and LOQ for determination. And we were able to establish a method to detect low quantities of NTPs in mRNA. This method also could be used to follow your consumption of NTPs during your IVT reaction. And this is an example of an IVT reactions that was done. And it's 0 to 30 minutes. And you can see in 30 minutes, quite a bit of the C was gone, the A, the U depletion, the G, and the depletion of the AG clean cap. So this method could be used for both in-process and release to monitor your reaction and release of the product. So in summary, it's the method is capable of separating and detecting residual NTPs, modified NTPs, and capping agent. Method is capable of in-process testing. The method is quantitative. We've shown specificity, clear separation between NTPs and clean cap. Linearity, concentration between 0.2 millimolar to 1.2 millimolar. The accuracy at 50, 100, and 150 nominal concentration of 0.4. Precision, the percent RSDs were below 5%. And the LOQ is at 0.04 millimolar for NTPs and 0.16 millimolar for clean cap. So it's actually providing to be a very good method to measure residual and to monitor and adjust your consumption of NTPs for your in-process while you're making your IVT. So in conclusion, control of critical raw material for making mRNA with proper battery of tests is very, very important. Advantage of CleanCap reagents, the manufacturing process of mRNA. Developed of quantitative test methods will continue to evolve to address the evolving regulatory requirement. We have at TriLink developed generic assays that can be optimized for specific mRNA constructs. These methods have been used and qualified for over 50 constructs for different customers that we do as a CDMO. So those methods right now went through completed different sequences for customers that have their own sequences, and we were able to to show that they could be qualified and adaptable for any construct. Use of IPRP method could be utilized for multiple aspects of mRNA characterization. We do have a robust gap in efficiency and poly-A tail assay, and an HPLC method to detect residual NTPs, modified NTPs, and capping agents. TriLink does, as mentioned earlier in the introduction, we do provide custom manufacturing capabilities, and this chart shows that we do provide mRNA capping through CleanCap all the way from discovery and research to commercialization. And also we do provide NTPs from discovery and modified NTP from discovery to commercialization. And the nice thing about that, if any customer have an idea of modifying certain NTP that they think it makes a difference, we have the expertise to make that. And if it works, we can scale it up and transfer it to GMP for their commercialization to be used in their construct. We do make oligonucleotides up to clinical phase, mRNA up to clinical phase, and plasmid DNA up to clinical phase from discovery and research. We are ISO certified facility, and we do have an annual ISO audit that, and also customer audits, and we support any audits that customers need to check our quality systems to do any business with us. With that, I would like to thank you for your time and open it to questions.
Okay, great stuff. Thank you, Khaled. And our audience, I'm sure, knows that TriLink is not a newcomer to mRNA. You have a long, deep history and experience with it. So that will be taken into account when listening to the context of what you're saying. It's a great review of mRNA manufacturing and the quality control landscape. And especially, thank you for clarifying the sequence separation advantages over and above capillary electrophoresis. We got a lot of questions in. I looked ahead, and a lot of them actually are quite detailed. And we won't have time to answer all questions. So the ones we don't get to, we'll answer offline. And we may keep many of the questions a bit more general because some of the questions that came in are very, very specific and perhaps not as interesting to every member of the audience. Before we get started on the question and answer session, I'd like to remind the audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found directly below the video player. And the question I'd like to start with is, does TriLink offer analytical services?
Yes, we do. And we are right now onboarding an additional 2,500 square feet that's dedicated for analytical services and analytical characterization of mRNA. The company has invested heavily into adding more equipment, and and we do provide that, and we do have the capability that presenting. So most of the methods that we test are in-house. We hardly farm any methods out.
Terrific. And I don't know if you know this, but is that more based on the raw materials, or is it more based on mid-process requests that you're getting currently?
Right now, mainly since we do have CDMO business for mRNA, the customers that are actually coming to us to make the mRNA, they are asking us to do all the analytical methods for them also. Wow. Because of the, of the basic methods that we've developed, the generic one that we develop and have them in place. So that kind of speed up the program.
Terrific. Next question, and you covered this to some extent. Are the analytical methods sequence specific?
Yes, they are. And we've shown not only it's sequence specific, but also NTP specific. So if you change the uridine to N-methyl pseudouridine, you're going to have different interactions with columns. You're going to have different interaction with the matrices that could impact some of the quantitation.
Okay, that sounds right. How does the length of the mRNA sequence impact the analytical methods that you're talking about? So I guess basically short versus long.
So again, if you look at So two things. If you look at IPRP method, the larger the molecule is, the broader the peak. So you'd have to do some more development in terms of the chromatography. And that's also seen in proteins. So if you make a very large protein, the chromatography doesn't look very pretty. But also we found that the interaction of the column with the sequence and length dependent, you really get different retention time. So it's really important to do your feasibility to understand where is your retention time and what things could change it. So that way you can have a robust method for release and stability.
Yeah, that makes sense. I'm a proteomics guy, so I'm not surprised you didn't mention gel electrophoresis, but capillary electrophoresis does not seem to have enough horsepower for what you guys are doing. Okay. Next question. Are the assays that you just covered construct specific?
Yes, they are, because if you change your 5' UTR and the 3' UTR, you would have to design different DNA enzymes for cleavage, and it could have other impact on other assays.
Okay. Why is it important to test for residual impurities?
It is extremely important because that's one of the regulatory requirements. The regulatory requirements want to see clearance of everything that you put in that is not related to your product is out at the end of the process. So even by theory, we can say, NTPs are very small. I'm putting it on a certain column. Everything should go and not be in, it should be removed and not be in my product. The agency will still like this, they will still request to see a proof of that. And that's why it's important.
Actually, I did notice you talked about USP and not really FDA. So I don't know if you want to comment on that. It's kind of related to the way you set up your methods.
So the USP is, as everybody knows, they kind of set the standard for what is required to test, through their monographs and all the chapters for testing requirement. So I mentioned the USP because they kind of pulled a lot of stuff together to put a chapter with all the battery of tests that could be used for characterization of mRNA. And the regulatory, like the FDA, is more about filing, not necessarily what methods you use. So there's a distinction there.
Yeah. No, I'm glad I asked, because I do know the distinction, but I'm wondering in practice. But the next one is kind of somewhat related. What are the main hurdles to analytical methods development and qualification?
Specificity and sensitivity. Those are, so we rely a lot on LC-MS because that could provide a lot of information that just an HPLC chromatogram or a CE would not tell you. So right now, to understand what's going on with your product, you really need the sensitivity and the selectivity.
Yeah, I'm with you on that. So again, somewhat related, what exactly is 120 and 80 indicated, I guess, from your slide earlier, length of the poly A tails?
So this one, we just wanted to show that there's no concept behind it. We just chose 80 and 120 just to see if we can see a separation with the same sequence that have different poly A tails. So that way, if someone by mistake made a shorter poly A tail for some reason, and you have your method, qualified to measure your actual product, you'll be able to see that there's something wrong with your poly A tail because it could have a different performance in the cell regarding the translation.
Yeah, actually, does that go to what you were talking about, keeping some of the methods consistent so then you can understand the process better?
Correct.
You mentioned.
Yeah.
All right, moving on. For the untailed mRNA separation, what mRNA is used?
That was, I believe, the wild type fluke catalog mRNA that we have. We have it as 120 tail, and we made it without a tail just for that purpose, small scale reaction. So we can study that, the method regarding that.
Thank you for that. That actually helps. So I will ask a couple of more specific questions since we do have a little time. So, which LC method and ionization technique was used for the LCMS experiment? IPLC should not be ESI compatible. Is that correct?
Correct. We've used reverse phase method. So the LCMS is a little bit different than the IPRP method. The IPRP method is strictly for HPLC. The chromatography conditions for the LCMS is a little bit different, and that's correct. You need to do that for ionization. And the mass spec that was used is Thermo Fisher Orbitrap.
I want to give props to Joey Tarantino for doing a good job triaging these, because the next question is, what are the main impurities resolved by the IPRP method?
I did not show a lot of that, but you can see a lot of your aborted sequences and to see that you cleared them all. And also if you have an untailed. So those are extremely important, because if you do not detect your, to make sure that you got rid of all your aborted sequences, you're going to have a lot of impurity, even though they will not translate, because most likely your aborted sequences does not have the poly A tail or the capping agent or both. They will give you the wrong concentration and all of that. So this is a huge advantage, especially, to ensure that you have an ultra pure mRNA for your product.
You're doing a good job fielding these. They're not easy. So the next one is a bit more specific as well. Have you tried to use HPLC to do capping efficiency quantification? And which column is best for DNazyme digested capping quantification?
We use the Thermo Fisher DNA pack column for most of these methods. And sorry, what was the beginning of the question? I did not.
Yeah, hang on. Let's. Have you tried to use HPLC to do capping efficiency quantification?
So does that mean have you used it with the UV instead of LCMS?
I would guess as a triage phase to then move on to either another more quantitative method.
We look a little bit at the UV. You really cannot separate. I only saw one method that is done, I think, by either Pfizer or Moderna, where they use magnetic beads and they design specific biotin oligos in order to do a UV quantitative method by HPLC. I know there's Sartorius does presentation for their in-process. I've talked to them about that. That method have not been qualified, and they don't recommend it for release.
So great answer. You're doing extremely well, because they're not easy to answer on the fly. We're running out of time, but there are a couple of other interesting questions. What percentage purity do you consider an ultra pure mRNA?
That is a very good question. Depending on the method, if you go with a frag, you don't want to use HPLC. The larger you get, so if you get close to, let's say, 9,000 NTP, you're pushing the limits at 70% because of the smear analysis. By HPLC, that could be close to 70. If you go to a shorter mRNA, which you have better chromatography and your CE with your frag analyzer look better, you start seeing in the 80-85% pure.
Nice, thank you, Khaled. So this may be the last question, and I'm asking because I'm partly curious myself. There's a bit of a debate on the biology of this. So any regulatory requirement on plasmid raw material, purity, impurity, identity, etc.?
Okay, so that is a lively debate. I've been involved in quite a few. It's a really good question, and I'm not going to skirt it. I'll just go straight to the point. Plasmid is also used for antibodies to make your material. Is that correct?
Yes.
Okay, so the regulation for that is very well specified. So why that would be different for mRNA?
Yeah, so I've been part of that debate as well. And I think it's because there's a speculation that empty plasmids can block enough of the host's innate response to then give the more full packages a chance to do its job, I believe is the main response to what you just said.
That's a true statement, but do we have the evidence? We have to show an experimental evidence for the agency to say yes, because right now I've seen a lot of people talking about plasmid GMP-like. I never heard of that. You either have GMP or non-GMP. So,
well, it's actually part of the charm of being in a product line and industry where things are just beginning to emerge. So it's
correct.
It's a healthy debate. Well,
correct.
I've very much enjoyed your presentation, especially since you set the context and explained a lot of the fundamentals. So thank you, Khalid. But unfortunately, we're out of time. Any last thoughts or feelings, statements from you?
Thank you for everyone, and I do appreciate the questions. It's I think it was very, very well oriented. Thank you.
Yeah, there was a lot more questions we didn't get time to, so don't fret. We will answer them offline.
Sure.
So thank you, Khalid. We are out of time. I want to thank the audience for attending and for participating in today's event. And I'd also like to thank our sponsor, TriLink Biotechnologies, for making today's educational webcast possible. Before you go, we'd like to ask everyone in the audience to participate in a very brief survey. The survey will populate automatically after today's presentation has ended. You'll receive an email alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thanks again, Khalid. I'll look forward to seeing you in San Diego later this month.
Thank you.
Thanks to everybody for joining, and we'll see you next time. Thank you and goodbye.